In this video, we'll discuss about spectrophotometer. What is spectrophotometer? This is an instrument commonly used in laboratory. How we'll define that spectrophotometer? It is an instrument that measures the amount of light observed by sample. So it measures the amount of light observed from the samples. The light observed by the sample is directly proportional to the concentration of the sample in the solution. So concentration increases means observation of light will increase. Light observation is directly proportional to concentration of that sample. Next, principle of spectrophotometry. Spectrophotometer technique are mostly used to measure the concentration of solute in the solution. Solution here our test solution by measuring the amount of light that is observed by solution in a cuvette placed in the spectrophotometer. So, spectrophotometer measure that amount of light observed from that sample. The concentration of solute directly proportional to the amount of light observed from the sample. Their concentration are high means that observation of light also high. So this is the way we measure our sample. Now the working of spectrophotometer: light source, lens, monochromator, our samples, detector and digital display or meter. These are the important part of instrument. What are the things? Light source, lens, monochromator, sample, detector and digital display. First source is light. Light source, it produces a desired range of wavelength of light. Second one is lens. Collimator transmitted a straight beam of light. It will transmitted straight beam of light coming from the light source. So it collect all the light source to the monochromator. So this lens will collect all the light from to monochromator. What is the role of monochromator? Separate light. It split the light into its component wavelength. Based on that wavelength, it will split. That means input is polychromatic, output is monochromatic. Now we see only one wavelength of light. Now it is transmitted to the sample solution. Any photosensitive device are used as a detector of radiation energy. Here we are using photocell. This one is the simplest photo detector. Then display device, the data is formed. The detector and displayed by the readout device such as analog, digital display. Output also transmitted to the computer or a printer. From here we can see our result, that is how much light is observed from the sample. So this one is the statement form: source, light source, then lens transmit a straight beam of light, then monochromator. What is the role? It split that light, that means polychromatic to monochromatic. Here monochromator, maximum of the laboratory there are using prism. then detector. Finally, application of spectrophotometer. This spectrophotometer we can use in clinical chemistry, molecular biology, immunology, hematology, along with the detection of concentration of substances, detection of impurities, then monitoring dissolved oxygen content in freshwater and marine water ecosystem, then characterization of protein, detection of functional group, that aldehyde group, keto group, methyl group, like that, then respiratory gas analysis in hospital, molecular weight determination. We are using this spectrophotometer. This one is the notes format of spectrophotometer. Thanks for watching.